Thank you for asking me for a presentation. So today I'm going to talk about one of the projects in my lab. That is to the role of MAP kinase phosphatase, also called the MKP1 or DASPER1 in cisplatin resistance. So this project I've been working on for quite a while. So I try to get the funding. At one time point, it's almost close to the funding, but that's how it works, when we write the grant. So before I get into MKP1, so I'd like to refresh your memory what is the MAP kinase pathway, because the MKP1 acts on the MAP kinase to regulate the cell signaling pathways. So as you know, in mammalian cells, there are three major MAP kinase pathways, namely JNK and P38. So growth factors can activate which can lead to cell growth and proliferation. So on the other hand, stimuli like stress can activate JNK and P38, which can lead to many outcomes. One of the outcomes is to cause apoptosis. So in cells, there are the growth proteins called the MAP kinase phosphatase. These family members which can remove phosphor groups from JNK and P38. Because phosphorylation is critical for the activation of MAP kinase. Therefore, when phosphor groups are removed from MAP kinase, which can inactivate MAP kinase. So there are 11 members of MAP kinase phosphatase. So MKP1 or DASPER1 is a founding member of the MAP kinase phosphatase family. So it can be induced by stimuli, including mitogen or stress, including the oxidative stress. So what MKP1 does is to selectively dephosphorate both phosphorthreonine and the tyrosine residues of this called the TXY motif in the MAP kinase. So dephosphorylation of TXY motif in MAP kinase is absolutely required for the MAP kinase phosphorylation. So in terms of the substrates, so the dogma is only all three JNK, P38, and are substrate of the MKP1. But JNK and P38 are better substrate over the so if three MAP kinases are available, so MKP1 first dephosphorate the P38 and JNK over the The role of MKP1 in cancers, when we start this project, about 10 years ago now. So that time it was shown that the MKP1 expression is altered in a number of cancers, including breast, ovarian, today I'm going to talk about. So I use auto because in some cancers, which is overexpressed, some are downregulated, but most of cancers are overexpressed the MKP1. And in the pancreatic cancer models, if you downregulate MKP1, you can suppress the tumor genicity. And when MKP1 is overexpressed, it inhibits the apoptosis. The time when we start with this project, the role of MKP1 in chemo-resistant, particularly, specifically in a cisplatin or platinum resistant, is not known. So first, we ask the question, what happens if we overexpress MKP1? So here we took the ovarian cancer line, we stably express MKP1. Show here, this is exogenous. And then we look at the cell survival. So here we use colony formation assay. As you can see, the treatment we use 0.1, 0.5, which is pharmacologic achievable in patients, in clinically. So we show that overexpressed MKP1 can increase colony formation compared to control vector cells. Then we ask another way around. So we took ovarian cancer line, we knocked down by shRNA, and then we look at what happened if we treat with cisplatin. And also, we show that this MKP1 can be induced by cisplatin. So here we knock down MKP1, which cannot be induced by cisplatin. And in cells, when MKP1 is knocked down, we can increase part cleavage induced by the cisplatin, but it's not as obvious as in the in control cells. Again, we show that in the opposing result. So we show when we knock down MKP1, we can eliminate colony formation as compared to the vector controls. Now we establish the expression of MKP1 with the cisplatin resistant. This is in the cell model. Then, because this is an ovarian cancer group, and all we know that ovarian cancer patients is not primary resistant, it's acquired resistance is clinical problem. So what we did, we took this ovarian cancer line of OV433. We start with very low dose of cisplatin, then we slightly increase dose over the period of 6 months. We establish the resistant line, show here use colony formation assay. And also we show that in a resistant line, the cells express high level MKP1 compared to parental cells. Because all these cell line can be artificial, then what we ask if this is clinical relevant. So I collaborated with one of my friends, so we got a number of patient samples. So unfortunately, we only get 2 patient samples, which we have primary tumor and also recurrent tumor. So we look at the MKP1 and the phospho-MKP1 in both primary and the recurrent tumors. We found that in recurrent tumors, MKP1 and the phospho-MKP1, which is a more stable MKP1, the level is higher in recurrent compared to parental, and in both patients. So those patients treat with platinum-based chemotherapy. After 10.6 months, another close to 11 months, and the patient developed a recurrent tumor. So when I included these two samples in my grant application, so I always criticize by reviewers say, okay, you only have 2 patient samples. As you know, it's extremely challenging if you can get primary tumor and recurrent tumor from same patients. It's extremely challenging. But I mean, if somehow I can get more, and then maybe I can strengthen this preliminary data. So then we went to a TCGA database. We look at the series of ovarian cancer patients who are treated with platinum-based chemotherapy. Then we look at the level of MKP1 versus the poor progression free survival time. So here shows that the patients with high level MKP1 had worse poor progression free survival compared to patients who had lower of MKP1, which has better PFS. Now we establish the based on this slide and the previous few slides, we establish the level of MKP1 and the cisplatin or platinum resistant, and also the worse PFS. Then, so this is just briefly this part. MKP1 inhibits cisplatin-induced apoptosis to promote the resistance, and then we show that the level of MKP1 is increasing cisplatin-resistant ovarian cancer cell lines and the recurrent ovarian tumors compared to their counterparts. So suggests MK1 may be a therapeutic target for ovarian cancer. Then question is how to target for improve cisplatin-based chemotherapy. So of course, the direct way is we can develop the small molecule to target MKP1. As you know, so currently everybody target the kinase, but phosphatase is much difficult and it's very challenging because there are overlap of substrates, and also you start with like level is not that high, then you want to decrease even lower. I mean, practically and technically it's pretty challenging. So today of course we have a project which we try to have small molecule which you can target DSP1, although it's not very effective kind of now. We try to improve that. But today I'm going to talk about how we can target based on mechanism. So this is what we start with. So when we have this resistant line and we treat with cis5, we see when we overexpress MKP1, we found the top level, which is higher than vector control, and also corresponding PARP, which is functional PARP. And here just show you the high level MKP1, it's more resistant to cisplatin compared to vector control. And conversely, we also show that when we knock down MKP1, we have less PARP level and the PARP, and also more sensitive to cisplatin. Now we establish high level of MKP1 and more cisplatin resistance. Because as I mentioned, the dogma is MKP1 only target the MAP kinases. So then we ask this effect mastered through the MAP kinases. So first we like identify which MAP kinase pathways is involved in this process. So we had this MKP1 knockout MAP cells, embryonic fibroblasts. So plus and the minus. So first we verify if those cells are behavior same as ovarian cancer cells we manipulated. So again, we show that knockout cells has low level of PARP, and also increased cleavable PARP when we treat with cisplatin, and also PARP also lower compared to plus cells. And when we treat with cisplatin, we can see the increased activation of all three MAP kinases, PARP, JUNK, which we use CJUNK as the radar, and also P38 we use CCRAP, and also you can see here is ERK activation. Although we can induce these three MAP kinase pathways by cisplatin, but the level is not as high as MKP1 knockout, and similarly we see the minus cells high is more sensitive to cisplatin. Then we ask which the pathway is involved in the regulation of PARP1. So initially we used inhibitors. We used three different kind of inhibitors for SP, which is a JUNK inhibitor, U0126, which is a MAC inhibitor upstream of and SP, which is P38 inhibitor. We treat MAP cells with cisplatin in the absence or in the presence of the inhibitor. And the idea is if it's involved, we can rescue the cell death induced by cisplatin. So basically, if we add U0126, increase cell death in the MAP cell, in the minus MAP cells, so which we exclude the involvement of the ERK. And then we show that the SP, JUNK, and the P38 can partially rescue, but when we look at the level of PARP1 and the PARP, although SP, which is P38, can slightly rescue this phenotype, but the SP JAK inhibitor is more obvious compared to SP. Then the SP, then we thought this is probably JAK plays major role in this process. So here we took MAP cells we treat with SP in the absence or presence of cisplatin. We show that actually in both MAP cells we can see when we cisplatin induce the JAK, and if we add the SP, and we can see decreased level of PARP1 and also decreased C-jun phosphorylation, which indicates that actually the pathway is inhibited by JAK. Then so we got knockout MAP cells from JAK knockout MAP cells from Roger Davis, and then we also look at the cells. If JAK is really important in this process, so we should expect that there's more PARP and higher PARP and the PARP1. So this exact the case. So you can see. So in the absence or presence of cisplatin we treat, we can see here just the knockout cells. There's no JAK, which is it's expected, and the high level PARP1 and the high level PARP compared to the wild type cells. So these slides. So step the correlation. Actually the JAK is important for the PARP expression in the context of MKP1. Then we ask what's the mechanism. So one thing you could think about is maybe the JAK is phosphorate the PARP1, then lead to the degradation. So what we did here, we knocked down and the MAP and the knockout MAP cells. So we look at the ubiquitination of PARP1. So as you can see in both ovarian cancer knockdown cells and in MAP cells, which MK1 is knocked out, we see the high level of ubiquitinated PARP1 compared to the either wild type or the non-targeted shRNA cells. And also here shows that the level is, if MKP1 is knocked down, we can see the low level of PARP1, and also here is low level PARP as well. Then we use the inhibitor, which is a JAK inhibitor. In the MAP cells, in the absence or in the presence of SP inhibitor, we add inhibitor. We can decrease the ubiquitinated PARP1 in both the MAP cells, and when we inhibit the JAK, we can see the increase partially can rescue the PARP1 level, and here just show you the SP. Actually, it totally inhibit the C-jun phosphorylation, means the inhibitor works. Then we did further. We took the this knockout ovarian cancers. We used shRNA to knock down JAK or use the control shRNA. So when we knock down the JAK, we can decrease the ubiquitinated PARP1, and also corresponding we can increase PARP level, total PARP level. So suggest that actually JAK actually it's play a role in ubiquitinate in the PARP1 ubiquitination. So then we took knockout MAP cells from JANK1 to knockout mice. We look at, we did similar IP. So as you can see, so in the minus cells, we detect much less of ubiquitinate the PARP1 compared to plus cells. So here shows the input of minor cells have high level of PARP1. So now we establish that MKP1 may inhibit the JANK to impact PARP1 ubiquitination. Then we ask, because the MKP1 inhibits the JANK can be phosphorate the PARP1. So the idea is maybe this impact the phosphorylation, then phosphorylation can talk for the ubiquitination and the degradation. So what we did here, we took MAP cells, we treat with cisplatin, then we IP with the PARP1, then we look at the phosphoserine, because we don't know which residue can be phosphorylated by JANK in the PARP1. So as you can see in the cell treat with cisplatin, we detect a high level of phosphoserine in the minus cells as compared to plus cells. So similarly, in JANK1 to knockout cells, we detect the a high level of PARP1, but we did not see the phosphoserine minus compared to plus cells, which we believe that so interaction between the JANK and the PARP1, which can impact by MKP1, promotes the phosphorylation of serine phosphorylation of PARP1, which may lead to the ubiquitination and the degradation. So what this mechanism, how this mechanism can apply to the cisplatin resistant. So here we have this resistant OE433 cells parental resistant. Then we look at the so here I already show you that resistant cells have high level MKP1, and then also we see the high level PARP1 and also PARP1, and also we found that we treat with cisplatin, we can activate JANK, but activation of JANK in resistant lines much less than parental cells. Then we took resistant line, we knock down MKP1 or PARP1. Then we look at the MKP1 level, PARP1 level, PAR, and the JANK. So when we knock down MKP1, so we see the decrease the PARP1 level and the PAR level shown here, particularly when you treat with cisplatin. And when if we knock down PARP1, it doesn't affect the MKP1, which suggests that MKP1 is upstream of PARP1. And of course, you knock down PARP1, you can see the less PAR, and also you can see the but it doesn't affect the JANK, then suggests that JANK is upstream of the PARP1. And also we look at the cisplatin sensitivity. So here show you, we knock down MKP1 or PARP1, we can increase cisplatin sensitivity. So lastly, we have this overexpression MKP1 cells. We did, this is a very preliminary data. So we treat cells with cisplatin, of course, as with MK1 overexpression one, we see the increased cisplatin resistance, but we use the PAR inhibitor. Here we use ABT888, which the concentration we use it doesn't affect the viability. When we combine both, we can see the increase the cell death. Although ABT8A is not the, I believe it's not currently approved for treatment of breast cancer, ovarian cancer in patients with BRCA1/2 mutations. So basically, this started about 10 years ago when we started this project. So PARP inhibitors was not improved for the ovarian cancer treatment, but as we progressed this work, and then the PARP inhibitor was later on approved for the patient treatment. So that's the novelty now becomes an issue. Of course, you know, we have the if we can use this concept to treat ovarian cancer patient with MKP1 overexpression population, that'd be good. So this part I'm going to summarize. The MKP1 levels are correlated with PARP1 level, and the MK maintains the PARP1 level by inhibiting JNK-mediated PARP1 phosphorylation. We believe subsequently ubiquitination and degradation. So this study suggests that MK-mediated cis1 resistance can be bypassed by PARP1 inhibition. So this is the model we proposed. So we think when we treat cells with cis1, this is not only in ovarian cancer. We believe this can apply to lung cancer and also the cancers which are treated as first-line chemotherapy, platinum as first-line chemotherapy. So we think treatment with cis1 can activate JNK, which also can induce MKP1. So the outcome depends on which is more predominant. So when JNK is phosphorylated, it can phosphorylate PARP1. When PARP1 is phosphorylated, it tends to be ubiquitinated and degraded, which cells more easy to undergo apoptosis. On the other hand, if PARP1 is not phosphorylated, it becomes more stable, and also it tends to be more easy to survive, and also the cisplatin resistant. So this work, you know, I always miss one of my former assistant, Joanne Wang. So she did a lot of work in this on this project, and also, so I have, of course, I have some fundings, like initial funding by Party Foundation. Then later on, I use other funds to support this, including my NIH R1 and R21. But if I don't get the funding on this project, although I love, you know, this project, but I don't know how long I can hang in there for use other money to support the project, which seems have not get funding yet. So I'm going to stop here. Then